All right, it's 10:30. Welcome back to Soil 205. The plan for today is to continue our discussion on this whole electron potential. It is this PE, the potency of the electrons, which leads us directly to respiration. And the order listed here is actually wrong. I want to do the respiration piece first. Then we'll do a short break and do the fishbowl today. I hope that Caleb, and I forgot who the other person was. Fantastic, thank you for being here. So that we'll do that in the middle, and then we'll do the quiz review with the remainder of the time. That's the big idea. And just as a recap of what we did last time, which perhaps was a bit much. So we've reviewed acidity, right? So we've looked at high proton activity, low proton activity, and reminded ourselves what the pH scale was all about. Right? And we identified what we already knew, that there is a pH neutral, which is pH 7, right? That's right along this axis here. Right? And then we've added something new to it to say that, well, there's not only the activity of electrons, protons to consider, but there's also the activity of electrons to consider. And that added a new axis to it, and the neutral for that is about 470 millivolts. So somewhere around here is our arbitrarily defined neutral condition. So if we go north of this, that means we have a lower electron activity. And so things at this end are going to be more intense or have a tendency to accept electrons, versus if we go to this end with a higher electron activity, we are more going towards a tendency of the ability to donate electrons. So the reason to introduce both of these axes together is because they always work together, right? They don't work independently of each other, which maybe is what the textbook applies to you, but the changes in activity of protons and electrons always go together. And on this little graph, you sort of see where, sort of soils fall within this realm. And you also see sort of the reminder of what I pointed out just before we closed last time, that most things tend to happen along this axis here, to where if we go to a more alkaline conditions, we also go into conditions with a higher electron activity. And then the opposite, if we go to more acidic conditions, we go towards a lower electron activity. And which one is the chicken and which one is the egg here is kind of hard to tell, but most likely it is this axis here that is the chicken, and this axis here is the egg. So that's what we did last time. And what I want to do today is to put this in a maybe more easily digestible context. And that is to talk about respiration, or the process of how energy is being recovered from carbon compounds within the soil ecosystem. Okay. So with that, here's our soil ecosystem. We've got plants, we've got a soil surface, and we have the thing that drives it all, and that is the sun up there. And maybe what we need to remind ourselves of, that all living things need a couple of things. And what they need is a source of carbon, they need a source of energy, and they need to have some building blocks to do something with it. So let's call those building blocks nutrients. So not every life form is going to acquire them in the same way. There's one life form, and that's the plants and algae that we call photoautotrophs. So the photoautotrophs are the ones that are going to get their carbon from the atmosphere. So they take the CO2 right from here, and they can assimilate that carbon from CO2 using photoautotrophs, photons, the energy from the photons, to do this all. And where do they get their nutrients from? Well, in this case, they're going to get them from here. So that's all fine and good, but that's not how I function. And I assume you don't function that way either. So we fall into a large group of animals that are called chemoheterotrophs. Okay. And chemoheterotrophs, well, that is all kinds of animals. So here's my rendition of an animal. There is us. And I'm already implying here that the animal eats stuff that the photoautotroph produced. Then we may eat the animal, and then our feces and remains are then consumed by all kinds of little bugs in the soil. They're obviously not drawn to scale. Okay. So us, the microbes, are chemoheterotrophs, which means that we get our carbon from our food. The CO2 that is in the air is not a source of carbon from us. We need to get it from our food. We also need to get our energy from our food. And lastly, the nutrients also come from the food. So we are dependent on photoautotrophs to be the beginning of the food chain there to provide us with everything that we need to have in this kind of system. Okay, so this is the chemoheterotrophs. And then, of course, all of this works together in a big cycle, because the carbon that was assimilated by the photoautotrophs, including the energy and the nutrients, gets now cycled to the animal, the person, the microbes, and the microbes and everything in between are decomposing things and making, oops, I should have drawn this right, to the nutrients, and are making nutrients available again in this kind of system. So there's a couple of new terms to add to this, and that is that the nutrients that are stored in the organic material and the nutrients which are then available again for plants to be assimilated again, which really means that they are available in solution, or they are, let's say, they are mobile in the environment. If we go from the nutrients stored in organic matter to these nutrients that are mobile, we call that a mineralization. And if they are mobile and get assimilated by the plant, then we call this an immobilization. Now, I don't know about you, immobilization makes sense to me, right? They're soluble nutrients, they become immobile when they're assimilated, but mineralization makes very little sense to me, because when I think of making a mineral, I think of making a rock, but that's not what that is. But those are the terms, okay? So that's sort of the big context. Now, of course, this big context happens in terms of redox reactions. So we've looked at photosynthesis at some point in time. So now let's look at the opposite of it, the respiration. Okay? So let's go back to the equation that we already know by heart, C6H12O6 plus 6H2O. And the C6H12O6 there, that's sugar, and that's just going to be sort of my example for a carbon structure. Of course, we eat other things other than sugar as well. Sugar is the easiest in terms of writing it out as an equation, but this can be any kind of carbon structure, even things that are really difficult to digest, like lignin, for example. Okay. So the carbon structure there, the sugar there, is the one that provides, that has, or it doesn't provide, but has the stored energy within it. So there's all these carbon bonds. There's it has a high electron activity, and it's stored in that way there. Okay? So the sugar oxidizes to 6 CO2, and in the process frees up energy. Maybe I should add this energy later in this process. Okay. So the sugar becomes the electron donor. Okay. And the CO2 is not the electron acceptor, but the CO2 is the oxidized form of that carbon. Okay. So where do the electrons have to go to actually free up the energy? Well, they need to have somewhere to be accepted. And the thing that works as the electron acceptor is the oxygen, oops, O2. And our oxygen will be reduced, so here's the reduction, to water, to our 12 H2O. Okay. So the electrons are moving from the electron donor to the electron acceptor. Okay. And let's say we're going to draw this in here like such. The O2 accepts it, but then what we get with is that we reduce the oxygen to water, and thereby we have the electrons assembled there. But it's not just the electrons that move. So there's 24 of them, but along with the electrons move protons. So there's 24 electrons and the 24 protons. And there you see again why the changes in pH and the changes in Pa go hand in hand, because these are going to be happening at the same time. Okay. So the H2O now here, of course, is just to add the label to it. This is the reduced form. And that's basically all that happens here. So the way I've written this out is called half reaction. So we have the oxidizing reaction on the top, the reducing reaction on the bottom. And just as a reminder, they always go together, right? Oxidation, reduction, they don't happen independent of each other. They always go together, and this is just a convenient way to write this out to really show, where the electrons and protons are moving in this whole process. That's the big idea, okay? So just as a reminder again, how do you remember which one is an oxidation, which one is a reduction? Remember there was this whole oil rig where it said oxidation is loss and reduction is gain. And gain of electrons. Okay. So very counterintuitive terms once more. Okay. So what I've drawn out here as respiration, well, this one is respiration if there is oxygen present, and we call this aerobic respiration. Okay. So that's what we do, and that's what the majority of organisms in soil will do as well, if there is oxygen present. Now, soils sometimes don't have enough oxygen present. We've looked last time at, what happens if you have too much water in soils that will block the diffusion process, which means that the oxygen gets used up quite quickly, and then something else has to serve as that electron acceptor. And what that something else is, maybe comes out in this awfully small plot. But let me talk to you in what we have here. I'll read it out to you, and then hopefully it will all make sense despite the fact that this is really teeny tiny. So what we're trying to do is we're going to try to gain energy by consuming carbon structures, and we want to get the most energy out of the process. And the most energy you're going to get out of this process, if you are really low on this side of the half reaction scale, and you're really high on the other side. Because what happens then is that you maximize that EH, that electron activity potential. Okay, so what I marked down there in the far bottom there is our little glucose, our sucrose, and then on the other side the oxygen is right here, right? So you see we have a big spread between our electron acceptor and our electron donor, which means that we get a lot of energy out of this reaction. So that's all fine and good. This is how we would like to have there. Let me point out some other things that are on here. So now in yellow, we're talking a lot about, fueling our cars now as electric cars, right? Making battery operated cars. Not too long ago, we were talking about hydrogen powered cars. And maybe that will come back because Toyota is still kind of on the, maybe this is what we need to be doing. So let's look at where this is, okay? So for a hydrogen powered car, we're going from this to oxygen, right? So from H2 to O2, okay? So lots of energy to be released in this process. That's why, they think that this will be one way to power our cars. Benefit, of course, would be that what comes out the tailpipe as a byproduct is, well, you combine H2 and O and you get water coming out, correct, right? So if this isn't working yet, well, not too long ago, and we're still doing this, we're putting ethanol in our gas tanks in small concentrations, right? So where's ethanol? Ethanol is right here, okay? So burning ethanol in the presence of oxygen, I mean, that's what happens in your combustion engine. Get a good bit of energy out of it, not too bad, okay? So, but what happens if oxygen is not present? Well, you're basically working your way down the ladder. Okay? So if there is no oxygen available for this reaction, next thing you're going to go to is to use nitrogen as an electron acceptor. If that's lacking, you're going to go to oxides like manganese oxides. If that's lacking, you just keep going down the steps until you're using sulfates, until you're using CO2. Now you're talking methanogenesis. But whenever you go down the list on this side here, you're getting less energy out of this reaction. So if you are in an environment where oxygen is present, and you as an organism want to go from glucose to oxygen, you're in good shape. If you're an organism that is capable of using nitrogen as an electron acceptor, and you're doing that, well, you get less energy out of that reaction, which means that you're kind of at a disadvantage. You're not going to be as lively as the one that used oxygen as the electron acceptor. But then when the oxygen is limiting, you're in good shape. So what happens then if oxygen is not available? So in the absence of O2, oops, that's not what I wanted. So the first thing that will happen is that our little reaction of our C6H12O6, which still goes to the reduced form of 6CO2, but is now going to use nitrate, NO3 minus, as your electron acceptor. And electrons are flowing the exact same way. And then you end up with NO2 minus plus 6H2O plus the energy, which I forgot to add to the previous graph. And it's the same thing. We have oxidation here. We have reduction here. We just used a different electron acceptor. Oops, acceptor. So this doesn't quite end there, because then the NO2 minus, oops, NO2 minus keeps reacting to where eventually you have N2 in the gaseous form plus 4H2O. And the downside of not having oxygen available and forcing a microbial life to use nitrogen as an electron acceptor is that this N2 in gaseous form is now leaving our system. So this one will diffuse out of the soil. And we call this process denitrification. Why is it of concern? Well, nitrogen was one of the main nutrients we need. One that where plants cannot use the N2 form, the dinitrogen form, and we had to have organisms like what we have discussed not too long ago, the ones that have a symbiosis with legumes, for example, to assimilate, to make that nitrogen bioavailable, or use lightning to add small amounts of bioavailable nitrogen to soils. So having waterlogged soils means that, well, now nitrogen gets used as an electron acceptor, and step by step, you're basically losing that nitrogen from your soils. So having waterlogged soils, especially during periods when the soil temperature is high and microbial activity is high, is a problem. Okay? So it doesn't, of course, end there. So let's say all the nitrogen now got depleted as well. So now you're just going to keep going down the list. So in order of preference, you're going to go O2, use some kind of nitrogen compounds. Then you're going to go to manganese, or the oxide forms, iron, sulfate, and lastly, you're going to go to methanogenesis. Oops, did we write this correctly? And your president just yesterday stated that methane was one of the areas of concern. He wasn't necessarily talking about methane emissions from soils. He was talking about the methane released as sort of leaks from hydrocarbon production. But methane released from soils is just as bad for the environment as any other source of methane. Okay. So this is the order of preference for microbes to use as electron acceptors. And it's an order of preference because as you go from left to right, you gain less energy as an organism. Okay, so it's bad for the organism in that sense. And then how it all relates back to the potency of the electron or the redox potential, this EH measure, is that the millivolt potential that we measure changes as we go from left to right. So the EH reduces, maybe this is a bad word to use in this case. So EH declines or decreases as we go from left to right. And we can use that to figure out where on this ladder of electron acceptors a soil may be. Right? So put non-polarizable electrode in the soil, measure the potential, and it reads 80 millivolts, and that will tell us, we are now using manganese oxides, for example, as an electron acceptor. Okay? That's the big idea. And that can be important, for example, if you're thinking about landfills, right? So you can measure that redox potential in landfills, and that will tell you, where things are in terms of decomposing in there and what might be decomposing in there. Or let's say you have some kind of pollutant spill, and you can actually map this on a larger scale, and then you can see based on that redox potential where your pollutant is at time and maybe how it is changing in terms of how it's degrading and also how it might be changing with water flow. So if you're thinking about a pollutant spill you could even, practical example, you have a dairy lagoon that you found out is leaking, and you're worried about all the effluent ending up in groundwater. You can actually map it with this redox potential. And you could see, how far away are we from groundwater? Is it degrading fast enough before we'll reach the groundwater? Okay? So has some practical uses there as well. Okay? So the other one is, this isn't another use, but another way of looking at the same thing. This is a figure from your textbook. So what we have here in this figure is that you're starting out with the soil that has plenty of oxygen available, and you're giving the soil a carbon source. Okay. So you have a carbon source, which, you see here, and then you're basically shutting off the oxygen supply to that soil so that the microbes within that soil will consume that carbon And at first, they're going to be using oxygen as the electron acceptor, which is why you see the oxygen concentration here, which is this line, decline with time. Okay? So oxygen is being used as the electron acceptor. They're gobbling up some of the carbon, which is why the carbon concentration here is declining. And at the same time, you see that there is a potential, this redox potential, which could be red. While there is oxygen available, you're at this kind of plateau. So you can read it, and then you would know, aha, there's plenty of oxygen available. Here's what's happening with my organic carbon. So now that the oxygen there is depleted, the next thing that gets used is nitrogen, okay? So then you see the nitrogen being used as an electron acceptor, so its concentration decreases. You have a lower redox potential, and your organic carbon, keeps getting gobbled up by microbes. But the rate of decomposition is going to decline because there's less energy available to do this whole thing. Okay? So up next, we're using manganese oxides. Okay, so now we're not gonna see a decline in something. We're actually going to see an increase in something, and that is Mn2+. So the soluble form of manganese increases because the oxides, which are not soluble, are being used as the electron acceptor. Okay? And in the same process, so carbon keeps declining. We now have a different plateau for the potential. Okay? So now that the manganese oxides are being used up, what comes next? Iron oxides. Same thing happening here. So we're going from iron oxides to iron 2+, and the concentration of it increases. And the rate of decline here of our carbon structure becomes less and less. Lastly, sorry, I missed up the manganese and the iron here. This was the manganese line. This is the iron line. And the one that I highlighted here saying that it is iron is not, it's hydrogen sulfate. And what happens here is that sulfates are being used as the electron acceptor, and the byproduct that comes out as the increase in concentration there is hydrogen sulfate. And then after that comes, then we're going to go methane. So methane concentration increases, which means now that we're using CO2 as the electron acceptor, and we are releasing methane in this whole process. Okay? So basically you're going through time, and you just see microbes going from one to the next to the next. And when I say microbes going from one to the next, well, they're different microbes. It's not going to be the same microbe that used to use oxygen as an electron acceptor that now uses nitrogen as an electron acceptor. No, it's a completely different one. Okay. So that's that piece. And now to the pretty pictures. Okay. So the redox potential can be used as a way to classify hydric soils, and hydric soils are ones that are waterlogged, or at least waterlogged most of the year. So we can, in addition to having the visual and the colors that go along with those, we can just characterize them based on the redox potential as well. And then here, one more picture, and it's the last one before the fishbowl. And what we're looking at here is a subsidence post. Okay. So you see that this is the University of Florida. So this is indeed somewhere in Florida. And what we have here, we're sitting on a histosol. Anybody remember what histosols are all about? Organic materials, right? And what makes the organic material pile up rather than decompose? Waterlogged, right? Okay. So what happened here with this histosol is that they drained the wetland, right? So they drained the wetland, and now what happens is that oxygen becomes available for the decomposition of the organic material, which previously was not available. And with that, your organic carbon that made up that histosol disappears in the form of CO2 into the atmosphere. So between 1927 and 2012, the land has subsided by that much due to the decomposition of the organic histosol with the introduction of oxygen. Okay.